Experiments were conducted in a Milpa, where 4 local land races of the common bean were planted together with the local weedy population. Molecular genetic markers, such as ISSR and isozyme, have been identified to distinguish wild and domesticated populations in this field. An analysis of these populations indicates that a relatively high percentage, 30 to 60%, of the wild and domesticated populations show genetic markers that may have been introduced by hybridization. Over the long term, gene flow between wild and domesticated beans may be sufficient to eliminate genetic differentiation between the two types of beans, in spite of the predominantly self-pollinated reproductive system. However, genome areas on the chromosomes around domestication genes display maximum differentiation between wild and domesticated beans, presumably due to disruptive selection. Also suggests that gene flow is very variable in its magnitude, as local factors play a predominant role, providing an additional impetus for breeding programs involving wild beans to broaden the genetic basis of domesticated common bean. Bean geneticists and breeders of the Milpa project have obtained promising preliminary results from selection in crosses between domesticated and wild beans. Progenies of these hybrids produce seed yields significantly higher than that of domesticated parent. Also in milpas, various types of squash can be found. In the Sierra Norte de Puebla, Cucurbita pepo land races are grown preferentially by Mazapa farmers. The methodology developed for on-farm squash breeding utilizes an experimental design area of squash intercrop with maize, generally 1 row of squash per 5 rows of maize. Pre-harvest selection pressure is applied at 50% and entails selection for such qualities as flower phenology, earliness, plant habit, for example bush type, and externally measured traits of the fruit, like size, ring color, and texture. Post-harvest selection pressure is applied at 50% and focuses on fruit and seed yield and fruit flesh quality traits, like color and or flavor. Evaluation of the populations include assessment of correlations among traits to assist in later breeding activities. For example, in one assessment of Cucurbita argiosperma, seed weight was found to be positively correlated with fruit weight, peduncle width, and fruit flesh thickness. Another parameter is optimal sample size, which helps determine the degree of variability within traits under selection.
Cosechamos todo y revisamos cuál es la mejor y las vamos partiendo y vemos la semilla que está mejor, qué peso tiene, todo eso. La criolla que había antes aquí, chiquitas, no era grande, era chiquita, pero después ya empecé a sacar la semilla y la empecé a vender y ya empecé a ver que había semilla más grande y ya me salieron más grandes las calabazas y ya esa semilla he venido echando.
Porque yo de por sí, desde antes que vinieron los señores, yo sembraba calabaza de por sí. Era más grande y más desabridona y más poca semilla, y ahorita los señores, pues ya lo que me vinieron a seleccionar, ya da más semilla. Más calabaza chica, pero ya da más semilla. No como la de nosotros que teníamos antes, no, porque pues daba poquita semilla.
With local farmer participation, the Milpa squash breeding team developed and released Phytomex STG, a new hull-less variety. Also, traditional breeding techniques are being used to produce herbicide-resistant squash and to incorporate disease and pest resistance in land races in cooperation with local farmers. This project, in collaboration with local farmers, seeks to reinforce the broad genetic diversity of cropland races, along with other useful plants growing in milpas. Marginal crops, like local land races of squash and beans, and neglected associates, for example, wild crop counterparts and calites of the milpa, have not reached their potential use due to a limited attention. The milpa is not only a multi-cropping system, but also a complex cultural structure that permits local communities to respond to unpredictable economic and environmental changes, as well as to experiment with new alternative lifestyles.
The modern corn breeder has a responsibility not only to improve the maize in the country in which he works, but also to recognize, to describe, and to preserve for future use the varieties and races which his own improved productions tend to replace and in some cases to extinguish. Yet, when it comes to developing a truly comprehensive science of science, it can be done only by comparing the generalizations derived from physical sciences with those of the biological and social sciences, and by attempting to integrate all three branches. I rather suspect that the raw material for such comparisons and for an integration is already available, and that it is only necessary that someone adopts this as an objective of his research.